Welcome to Chapter 18, Regulation of Gene Expression. This chapter fulfills objectives number 1, 3, and 4. Specifically in objective number 1, biological organization and feedback mechanisms, and in objective 3, cell structure and function and control. For objective 4, it's on heritable information in DNA. In this chapter, we're going to be covering the sections conducting the genetic orchestra, responding to environmental change, eukaryotic gene expression, differential gene expression in multicellular organisms, and cancer and cell cycle control. First off, on conducting the genetic orchestra, which is another way of saying gene regulation. You may be wondering, why do we even care? Why do we care about gene regulation? And why does the organism regulate its gene expression? Why doesn't it just express all of its genetics all at the same time? Well, let's look at two different examples that we're going to explore in more detail in the chapter. First off, let's look at our unicellular organisms over here, our bacteria. Now, let's say the bacteria have two different sugars in the environment. It could express its genes for metabolizing both glucose and lactose at the same time. But glucose costs less, so it makes sense for the organism to import and use glucose for as long as it can, because it gets more bang for its buck for less expenditure on its part. It just makes good economic sense as well as good biological sense. In the meantime, it keeps its lactose genes turned off. When glucose is gone, now it needs to turn on the genes for importing and using lactose. That's one of the reasons why unicellular organisms turn on and turn off genes or regulate their gene expression to better respond to their environment. Now let's look at multicellular organisms. Here we have two little mustard plants. We've got one that looks like a seedling. It's got some baby leaves here. It's got a baby root. It's got the middle part that we'll call the stem. The organism had to turn on genes in this area that express what are needed for the leaves. Down here it needed to turn off those genes and turn on root genes. When this doesn't happen appropriately, you get a glob of cells that don't quite know what they are. So for multicellular organisms to have different cells expressing different genes so that they can behave in different fashions, you need to turn on and turn off genes. You need to regulate gene expression. Let's start with responding to environmental change. Let's go back to our little bacterium who's needing to respond to different carbohydrates in the environment. There's two different ways to organize your genes, and I call this the phone list example. One way to organize your genes is alphabetically, like you see in a phone book. The other way that you can organize your phone list, and we see this on our phones, is by groups. We group our family together, we group our friends together, we group work together, etc. Now let's apply that example to genes and organisms. Eukaryotes have a tendency to use the phone book example of the phone list, where genes are placed on the chromosomes not necessarily based on function. Prokaryotes, on the other hand, use the grouping by function. Okay, so they group all of the genes necessary for lactose metabolism together on the same stretch of chromosome. Further on the chromosome, there's another group of genes that are used for making the amino acid tryptophan. We call these operons. Generally, we call them bacterial operons because we have studied them in bacteria. Now, there are two types of operons that you will be tested on. One of them is an example of positive regulation. That's the TRP operon. Now, I'm not going to go over that one, but it's in the book. Make sure you go over it, read it. If you have questions, hit that on the troublesome topics. I'm going to include a clip on the how a lac operon operates, and it's an example of negative regulation.